So before we talk about some of the specifics associated with cancer, I wanted to provide a bit of an overview. First, here are the hallmarks of cancer, so the attributes that all cancers have. By definition, cancer is uncontrolled cell growth. And what we mean by uncontrolled is that there is typically no need for the addition of growth factors. So we say that that is self-sufficient growth. In addition, cancer cells are insensitive to anti-growth signals. One we've heard about before is contact inhibition, but another is dependence on being attached to the extracellular matrix, or anchorage dependence. Cancer cells negate those things, so they are not inhibited by contact, nor does their division depend upon anchorage to the extracellular matrix. In addition to cell growth, cancer cells evade cell death signals, so they are less likely to undergo the process of apoptosis. This uncontrolled growth is generally due to genetic alterations, DNA mutations, but in some cases it's due to epigenetic changes. In addition to genetic alterations that lead to uncontrolled growth, additional genetic alterations typically lead to morphological changes, angiogenic capabilities, and what we mean by that is the ability to recruit blood vessels so that there's a vascular supply for nutrients and for the removal of wastes. In addition, cancer cells are invasive, so they are able to break through their surrounding tissue. Cancer cells are also immortal. The limit on normal cell growth is typically called the Hayflick limit, and cancer cells override that Hayflick limit so that they are able to divide indefinitely. That is an attribute typically attributed to stem cells, but cancer cells are in a way a form of stem cell in that they are immortal. Finally, cancer cells prefer the use of aerobic glycolysis to oxidative phosphorylation. We'll talk a little bit more about that in a minute. In addition to these hallmarks of cancer, cancer cells often add these last two things on the list in order to make themselves, let's say, more robust at growing and spreading. So genetic instability is one of the character traits often associated with cancer, meaning that the rate of genetic alterations that accumulate is faster or greater. And finally, cancer cells often recruit inflammatory cells, and while this may sound counterintuitive, cancer cells are capable of evading the detrimental effects of the immune system, and in fact they take advantage of some of those attributes of the immune system by recruiting inflammatory cells. Here are some other features of cancer, and I'm not going to read this list because we're going to talk about each of these characteristics or each of these features to some degree or another. This is just a summary of the things we're going to be talking about. So first, cancers are categorized, and cancers are going to be categorized by the cell that gives rise to them. So recall that we have three different tissue layers or germ layers that originated during development: the ectoderm, the mesoderm, and the endoderm that sort of derived from the primordial ectoderm during a process called gastrulation, where the outer part of an embryo sort of invaginates to become internal. Epithelial tissues are mostly derived of ectoderm, but there are mesodermal and endodermal epithelial tissues. Mesoderm is often associated with muscle tissue or connective tissue. Speaking specifically about the epithelial tissue, the thing that separates epithelium from mesoderm is connective tissue, specifically the basement membrane. The basement membrane is a specialized extracellular matrix comprised of a specific type of collagen as well as fibronectin and laminin. This combination of extracellular matrix proteins makes a very rigid basement membrane on which the epithelial cells sit and which separates the epithelial cells from the mesoderm or the connective tissue. Here it's labeled stroma. So again, cancers are categorized based upon the cells that gave rise to them. So epithelial cells give rise to cancers that we term carcinomas. And mesodermal cells give rise to tumors that we call sarcomas. Carcinomas are the most common, by far and away, the most common types of cancers. There are various theories as to why this is the case. One of those theories speaks to the rate at which epithelial cells divide normally relative to the rate at which mesodermal cells divide. So epithelial cells divide more rapidly, and it's possible that upon each division, a mistake or a mutation might occur, and because epithelial cells divide more often, the accumulation of mutations can therefore occur more often. Another theory as to why carcinomas are much more common is that epithelial cells are often the cells that are creating that lining. They're lining tissues, and linings are the cells that are going to be most frequently exposed to carcinogenic compounds. So carcinomas are subsequently divided into two different types of carcinomas. There are adenocarcinomas and squamous cell carcinomas. So recall that epithelial tissues essentially have two functions. They have, of course, that lining function or that barrier function, but they also have a secretory function. Some of the cells within every epithelial lining are responsible for secreting things. In the lung, that might be mucus, and in the gastrointestinal tract, that might be mucus or digestive enzymes. So if a carcinoma has arisen from some of those secretory cells, we call that an adenocarcinoma. If the tumor has arisen from one of the barrier-producing cells, we call that a squamous cell carcinoma. And this table gives you just a listing of the various types or locations of common carcinomas. Here is a table giving you a list of more common sarcomas, but I'm going to put more common in air quotes because recall that sarcomas are not very common at all. Sarcomas are tumors of typically connective tissue, and those connective tissues might be bone or cartilage at the top and the bottom of the list respectively, or it might be muscle cells or fat cells that give rise to the tumor. And all of those are listed in this table. A sort of subset of sarcomas, often separated from the list of sarcomas, are hematopoietic tumors. Hematopoiesis is the process of the generation of various types of blood cells, and we know that there are numerous types of blood cell cancers. These are often separated from sarcomas, even though blood is considered a connective tissue, because they have very different characteristics in that they are not solid tumors, but in fact overgrowing cells within the blood. Here is a list of common hematopoietic cancers, and you see that they are identified as leukemias and lymphomas. There used to be a distinction between leukemia and lymphoma in that leukemias were of the cells that are floating around in blood, and lymphomas were more solid tumors often associated with lymphatic tissue, but that's not always the case. The way that hematopoietic cancers are subdivided is based upon the stem cell that gave rise to it. So we have lymphocytic hematopoietic cancers, and we have myelogenous hematopoietic cancers. Lymphocytic cancers have arisen from those stem cells that give rise to T cells and B cells, and myelogenous hematopoietic cancers arise from the other sort of stem cell that gives rise to macrophages and the granulocytes. And I've identified what their cell of origin is here. So another general characteristic of cancers, in addition to being categorized, is that all of our cells have the capacity to become cancerous. And that is because cancer cells are using normal cellular processes in order to become cancerous. They're using the normal cellular tools that they have at hand. In some cases, cancer cells are misusing normal tools. So the tools, the proteins and the RNAs that are being used are normal. They're just not being used normally. So examples of misusing normal tools might be making too much of it, making not enough of it, or making it at the wrong place or at the wrong time. So that's misusing normal tools. Of course, we know that cancers are associated with genetic abnormalities, and some of those genetic abnormalities are within protein-coding genes. If those mutations give rise to an abnormal tool, one that functions abnormally, or one that doesn't function at all, so a broken tool, that would be an example of this abnormal or broken tool. So not misusing a normal tool, but having an abnormal tool. Cancers do result from genetic alterations. Some of those alterations are actually genetic, so changes in the DNA. In fact, most of them are, but there are instances where some of those alterations are epigenetic, so using the proper DNA abnormally. Sometimes those mutations are within protein-coding genes, as I said. Sometimes they're not. So it might be within, for example, a regulatory sequence like a promoter sequence or a intron or an intron. But we're going to focus on the DNA mutations or alterations that are in the protein coding genes. The genes that are typically altered in cancer fall into one of a few categories. Either they have a positive role in cell division. Alternatively, the same category could be a negative role in cell death. These genes are called proto-oncogenes when they are associated with normal cells. So if a gene has a positive role in cell division, this might be a growth factor or a growth factor receptor or one of the intracellular signaling molecules, the messengers that are conveying information from outside of the cell to the inside in response to one of the growth factors. Those genes are called proto-oncogenes. And when they become altered in cancer, we term them oncogenes. So we get rid of the proto. The second category of genes that are altered in cancer are those that have a negative role in cell division. Alternatively, it could have a positive role in cell death. It would be in this same category. These genes are termed tumor suppressor genes, and these are the genes that become broken or non-functional in cancer, and then we just call it a non-functional tumor suppressor gene. Sometimes this last category, the genes whose proteins have a role in the maintenance of DNA integrity, are contained within the tumor suppressor genes. I've chosen to separate it out because these genes are, the products of these genes are functioning specifically to maintain proper DNA sequence and proper DNA structure specifically. Cancers develop gradually. Nobody comes down with cancer. Cancer develops. It is a continuum. And that is because cancers result from an accumulation of a number of genetic alterations. A single genetic alteration will not give rise to cancer. So here we see the cancer continuum, starting at a normal cell. And when one or more genetic alterations develops, those normal cancer cells begin to overgrow. When those normal cancer cells begin to grow, it produces something called hyperplasia. So hyperplasia is the overgrowth of normal-looking cells. I'll point out that on the left-hand side of this continuum, I also have added the word metaplasia. Metaplasia is a word that's used to describe normal cells growing normally, but in an abnormal place. The most common example of metaplasia is a type of precancer called Barrett's esophagitis, where some of the stomach-ish cells have migrated up into the esophagus. So we have stomach-looking cells in the esophagus. That's thought to be a sort of precancer. The stomach-looking cells are not abnormal stomach-looking cells. They just happen to be in the wrong place. Barrett's esophagitis often, as I said, is a precursor to cancer, but it results from intense reflux. So we have normal cells that might be growing in an abnormal place or might appear in an abnormal place. That's metaplasia. And we have normal looking cells that begin to overgrow, and we term that hyperplasia. Examples of hyperplasia might be skin tags or a very common type of hyperplasia that one would see in the dental office would be gingival hyperplasia, so overgrowth of the gums, and that tends to sort of impinge upon where your teeth should be showing. But this is not cancer, it's just simply overgrowth of cells. Once those cells become dysplastic or looking different. We call it dysplasia. And dysplasia, again, is when cells just start to look funky. So dysplasia is often considered a form of precancer, in that the belief is that if a dysplastic tissue or dysplastic cells are allowed to grow long enough, they will become cancer. This is a controversial idea because there are some physicians who believe that dysplastic cells will not all become cancerous, and in fact we've never done studies to determine what percentage of dysplastic cells will become cancerous, because some people believe this is probably unethical to let something continue to grow if it might become cancerous. So on the right-hand side of this micrograph, we see normal-looking tissue, and what we see highlighted by those white arrows are some dysplastic cells or abnormal-looking cells. The dark purple in those abnormal cells is the nucleus, and we see that the cell itself, the dysplastic cell, is larger relative to the normal-looking cells. But in addition to that, the nucleus is actually quite dark and quite large relative to the normal-looking cells. And this is one of the attributes that pathologists are looking for when they see tissue samples like this. They're looking for that ratio between cytoplasm and nucleus, and cancer cells tend to be not only larger, but tend to have a larger nucleus. So we've moved from normal cells to overgrowing cells along the continuum, and now those overgrowing cells have accumulated additional mutations such that they begin looking a little weird. That's dysplasia, and that, as I said, is often considered a precancerous condition. It's not cancer. This would be a benign tumor. It's when we begin breaking through the basement membrane, and we see that highlighted just, oh, I can't circle it. Highlighted in the in situ cancer. And the in situ cancer is a malignancy. It is a neoplastic tissue, and the hallmark of a cancer is its ability to break through that connective tissue barrier, that basement membrane. That is one of the hallmarks of cancer, invasiveness, and this is what we're seeing in an in situ cancer. This is malignant, and it has invaded, but it has not become a metastatic tumor. The last thing on the last step on the continuum is an invasive cancer, and you see here in this diagram that this cancer has not only broken through the basement membrane, but it's broken through all the surrounding tissue and found its way into, in this image, a blood vessel. This would allow these tumor cells to move to various parts around the body, various tissues, various organs, and this would be what we would consider a metastatic tumor. Not all metastatic tumors move through the blood vessels. Some of them are able to do so through the lymphatic vessels. So this is an invasive cancer, and what we see here is the lack of a basement membrane. The basement membrane is identified in that sort of brownish color. On the right-hand side, I wish I could highlight this, but on the right-hand side, we see, oh, here it is, the basement membrane. That's dark brown, but we see the absence of a basement membrane on this side of the image. So the degradation of that basement membrane is the invasiveness that is a hallmark of cancer. So this would be a carcinoma in situ. Not shown on that previous continuum is a step beyond metastasis or metastatic cancer, and this is something called anaplasia. We can look at these cells and we can see that they don't look normal, but we can't tell anything else about them. We have no idea what type of cell these are or where they came from. Anaplasia is a word used to describe cells that have de-differentiated or undifferentiated so much that they have no distinguishing feature of any type of cell in our body. These are anaplastic cells, and they are on the far right of that continuum. This is not a good thing. I have this slide in here because this is another controversial topic associated with cancer. We do believe that the vast majority of cancers are monoclonal. What that means is that a single cell has accumulated a various number of mutations such that it has become cancerous, and it has overgrown, it has invaded, et cetera, et cetera. So a single cell gave rise to all of the cells within a tumor. But there is refuting evidence to this. We like that theory because it's the easiest theory to swallow, a single cell gone awry. But there is some evidence to suggest that that may not be true, that tumors may actually have many different cancer cells in them, because when we look at a tumor, it happens to be very non-homogeneous. Cancer cells make energy differently than normal cells, and I said this at the very beginning. So normal cells typically import glucose from the extracellular environment into the cytoplasm. That glucose undergoes that process of glycolysis, where pyruvate is produced from that glucose molecule. Pyruvate is shuttled into the mitochondria, enters the Krebs cycle, and we use oxygen to create ATP. That's the normal situation. In a cancer cell, there is a switch to a preference for something that's called aerobic glycolysis. So we still require oxygen, but we prefer that glycolysis pathway. We prefer to produce our ATP in the cytoplasm. What's shown in this image is the upregulation of the RAS pathway and the PI3 kinase pathway that we spoke about before, leads to an increase in the concentrations of glucose transporter, an increase in the concentrations of a specific enzyme along the pathway to pyruvate production. This increase in the concentration of these proteins leads to the increase in aerobic glycolysis or ATP production in the cytoplasm. As a result, there's increased lactate production, and that's very often observed in when you grow cells in tumor cells in culture. We see that they produce a lot of lactic acid, and they often turn their medium a bright yellow color. This switch from oxidative phosphorylation to a preference for aerobic glycolysis is termed the Warburg effect. The last thing I want to point out with respect to cancer is that they don't affect everyone equally. Certainly, cancers are associated with genetic defects, and some of the information that's presented in this chart might be a result of genetics, in that we see certain cancers having a higher prevalence in certain countries at the top of this list and certain geographic regions at the bottom of this list. As an example, we'll just look at skin cancer. It has a very high prevalence in Australia and a very low prevalence in Japan, such that the relative risk of developing skin cancer is 155 times higher in Australia than it is in Japan. Looking at liver cancer, there is a very high rate of liver cancer in China and a very low rate of liver cancer in Canada, such that someone in Shanghai, China would have a 50 times higher probability of developing liver cancer than somebody who lives in Nova Scotia. Now again, this could be a genetic predisposition because, hey, Australia is an island, and so maybe the people who live in Australia are more likely to produce offspring with people in Australia. The people in Japan, Japan's an island, they are more likely to produce offspring with somebody who lives on the same island, right? So these all could be explained by genetics, or they could be explained by environmental factors. Clearly, the environment in Japan is different than the environment in Australia. So we know that the environment does play a role in the development of cancer. Here are just a couple of the environmental exposures that we know to be associated with the development of cancer. Tobacco use is associated with a higher risk of developing lung cancer, but not only lung cancer, bladder cancer, kidney cancer, breast cancer, a number of different cancers. Similarly, diet, a diet high in fat, high in fried foods, low in vegetables, that diet is associated with a higher risk of developing certain types of cancer. There are also environmental factors that play a role either positively or negatively in the development of cancer, but we don't understand why this occurs. So for example, exercise is associated with a lower risk of breast cancer. How does that happen? We don't know, but we know that there is a correlation. We know that low circulating vitamin D results in a higher risk of developing breast cancer and a higher mortality rate with colorectal cancer. Why that happens, we have no idea. Note that coffee consumption results in a lower risk of liver cancer, so drink up. In any event, I guess I want to point out here that cancer is on some level a genetic disorder, because we do know that genes and the alterations in genes are associated with the development of cancer. But that doesn't explain everything, because environmental factors also play a role in the development of cancer. Sometimes we know that those environmental exposures produce genetic alterations. These would be the carcinogens that are well known. But in some instances, we don't know that there is a carcinogen involved, and this could be explained through epigenetics.